Hello students, today I am going to discuss about one more important topic, that is gluconeogenesis. Okay, what is gluconeogenesis? What is gluconeogenesis? Gluconeogenesis is nothing but formation or synthesis of glucose from non-carbohydrate derivatives. What are those non-carbohydrate derivatives? Which are not sugar, okay, which are not carbohydrates. From that source actually this glucose will be synthesized, right? This will happen, so when there is extreme starvation condition, at the time the glucose will extreme starvation, normally the glucose will be synthesized from the glycogen which is stored in the liver. Some amount can be stored in the muscle, but in extreme starvation conditions, the whatever the glycogen stored I am again repeating, normally glycogen is the main source for glucose. This glycogen will be stored in the liver and muscles. Okay, whenever during starvation, the glycogen whatever stored in the liver and muscles will be utilized, total will be utilized, and there will be no glycogen level at the time. So what will happen, it will utilize, it won't the second source is fats. Before fat utilization, there will be another source, that is non-carbohydrates, which will be utilized to synthesize glucose again. Okay, I'm again repeating, normally glycogen will be stored in the liver and muscles, that will be utilized to form glucose. Okay, when we take food, the excess amount of glucose will be stored in the form of glycogen in the liver and muscles. After some time, when we are not taking food, at the time this glycogen will be utilized to form glucose, that will be utilized by the cells to produce energy. But when we are in long term starvation, so for example, if you are not taking food for 18 to 19 hours, at the time what will happen, this glycogen will be utilized, total will be utilized, and there will be no more glycogen to synthesize glucose. At that situation, actually cells utilize non-carbohydrates to synthesize glucose. This process is nothing but gluconeogenesis, formation of glucose from non-carbohydrate source. That process is nothing but gluconeogenesis. What are those non-carbohydrate substrates? Okay, there are many non-carbohydrate substrates. One among that, the main non-carbohydrate substrates we are talking here, so those are actually lactate. Okay, those are lactate, glycerol, glucogenic amino acids, and propionyl coenzyme A. Okay, lactate, glycerol, glucogenic amino acids, actually near about 20 amino acids are utilized to form proteins. So among these 20 amino acids, all amino acids except leucine will be used were called as gluconeogenic or glucogenic amino acids, because these amino acids will participate in the metabolic pathway to synthesize glucose. Okay, except leucine actually it is a ketogenic amino acid. These amino acids undergo a metabolic pathway to synthesize ketone bodies, so that's why this leucine is ketogenic amino acids. Other than leucine, all amino acids are glucogenic amino acids. These amino acids undergo metabolic pathway to produces glucose. Okay, whatever the substrates we are talking, these are actually non-carbohydrate substrates. These substrates are non-carbohydrate substrates. These non-carbohydrate substrates synthesize glucose. This process is nothing but gluconeogenesis. Okay, gluconeogenesis is nothing but formation of glucose from non-carbohydrate substrates. Okay, this process is nothing but gluconeogenesis. So these non-carbohydrate substrates are lactate, glycerol, glucogenic amino acids, and propionic acid. When we talk about glucogenic amino acids, especially we will talk about alanine. Okay, alanine is the exclusively for glucogenic amino acid. This alanine amino acid will be exclusively for glucose synthesis. Understood? Mainly we will talk about alanine. So before going to the glucogenesis, we'll talk few important topics. Okay, so we already discussed about the glycolysis. Okay, this gluconeogenesis is little bit reverse of the glycolysis pathway, but it is not total reverse. It is not a complete reverse of the glycolysis pathway. There are in glycolysis pathway, when we look into that pathway, we'll find three irreversible steps. Okay, those are not reversible steps. That those irreversible steps in gluconeogenesis pathway that should become reversible. Okay, how that irreversible steps in the glycolysis will become reversible step? By using four important enzymes. So I am again repeating, the glycolysis gluconeogenesis actually a little bit reversible of glycolysis pathway. Understood? So when it should become reversible, when the steps in the glycolysis should be reversible, so should reversible, then only the neogenesis pathway also will be little bit reversible of the glycolysis pathway. If the steps in the glycolysis are not reversible, then gluconeogenesis cannot be a reversible of glycolysis. So so what we have to do, whatever the steps in the glycolysis which are irreversible, that should be reversible. How they will be reversible? By using these four enzymes. So again I am repeating, there are three steps. There are how many steps? Three irreversible steps that should be reversible. How they are reversible? They utilize four enzymes. Three reversible steps should be three irreversible steps in the glycolysis should be reversed by using four important enzymes. So we'll see what are the four important enzymes. So those are pyruvate carboxylase. I am repeating. So as I am saying this, pyruvate carboxylase enzyme. Remember guys, whenever you see any carboxylase enzyme, so it is undergoing carboxylation reactions. So remember, it need three things, that is A, B and C. Okay, it requires three things, that is A and B and C. Whenever you see a carboxylation reactions, it needs three things. What is A? A is nothing but ATP. Okay, it need biotin. B is nothing but biotin. Biotin is a prosthetic group. Okay, and it needs C. C is nothing but CO2. Since it is a carboxylation reaction, it need ATP, biotin and CO2. Okay, remember guys, whenever you see any carboxylase enzyme, so it is undergoing carboxylation reaction, it need ABC, that is A is nothing but ATP, B is nothing but biotin and C is nothing but CO2. First enzyme is pyruvate carboxylase. Next enzyme is pyruvate carboxykinase. Remember guys, when you talk about kinase, the enzyme is adding phosphate group. Okay, next enzyme is fructose 1,6 bisphosphate. This is the thing, fructose 1,6 bisphosphatase. I am not talking about bisphosphate, it is bisphosphatase. See, I already discussed ACE is nothing but it is breaking. What it is breaking? It is a phosphatase enzyme, so it will break phosphate bond. It will break phosphate bond. Okay, so when it will break phosphate bond, so whatever the phosphate it is attached, it will be released. So one next enzyme is glucose 6 phosphatase. Glucose 6 phosphatase is also one more enzyme. It will break phosphate bond. Okay, these are the four enzymes which will be used to make three steps of glycolysis to reversible. Okay, so here in the glycolysis pathway to become gluconeogenesis, 6 ATPs will be utilized. Okay, then only the glucose will be synthesized. Understood? So 6 ATPs will be utilized. Okay, source of glucose during, actually I am reading out this thing. I already discussed in the starvation condition, whatever the glycogen which is there in the liver and muscles, that will be utilized. And in the starvation condition, the second source is non-carbohydrates that will synthesize glucose. What will happen during starvation? The glycogen total utilized. So Gluconeogenesis is another pathway for glucose productions. How? From where the glucose will be synthesized? From non-carbohydrate substrates. Okay, that is the thing. Sites for glycogenesis is cytoplasm and mitochondria of liver and kidneys. Renal cortex, actually it is a kidney. Okay, so why I'm talking like why I'm saying this is mitochondria, because in gluconeogenesis some of the substrates which will come from the mitochondria, especially from the Krebs cycle. Okay, we will talk in the future slides. Okay, from the Krebs cycle we will utilize many substrates to form glucose. Okay, that's why the site of the gluconeogenesis is the liver and also mitochondria. In liver cell, cytoplasm and mitochondria of liver cell and also kidney, that is renal cells. Okay, sorry. So gluconeogenesis not actually total reversible of glycolysis. Most importantly, some irreversible steps in the glycolysis can be reversible by important enzymes. That's what I have discussed already. Some irreversible steps are there that will be reversible, that will become reversible by using some important enzymes. What are those irreversible steps in the glycolysis that should become reversible by using the important enzymes? First reaction is that phosphoenol pyruvate should become pyruvate. This is an important step in the glycolysis, where the phosphoenol pyruvate, by using pyruvate kinase, that should become pyruvate. Okay, this is an actually irreversible step in the glycolysis. Okay, to synthesize glucose, actually when we see the glycolysis pathway, the starting substrate of the glycolysis pathway is glucose. Everyone know that thing. Okay, from the glucose many substrates will be synthesized, so ultimately we will get pyruvate. Okay, in gluconeogenesis we have to reverse the pathway. So pyruvate should become phosphoenol pyruvate should become 2-phosphoglycerate, and 2-phosphoglycerate should become 3-phosphoglycerate, and 3-phosphoglycerate should become 1,3-bisphosphoglycerate. From this we have to go for glyceraldehyde 3-phosphate. Okay, from glyceraldehyde 3-phosphate it should enter it should form fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate should form fructose 6-phosphate. This fructose 6-phosphate should form glucose 6-phosphate. This glucose 6-phosphate should form glucose. This is the reversible pathway. Understood? So what we have to form right now, from pyruvate it should form phosphoenol pyruvate. But remember guys, actually this is an irreversible step. Okay, this is not reversible. Understood? This is not reversible. This pyruvate should not form phosphoenol. Okay, this pyruvate should not form phosphoenol pyruvate. Right, so how this pyruvate should form phosphoenol pyruvate, that we have to see. So how it will form? So we know in the glycolysis pathway this phosphoenol pyruvate form pyruvate with the help of pyruvate kinase. Since it is a kinase, so it will utilize for phosphate. From where we will get the phosphate? From ATP. Sorry, here actually phosphoenol pyruvate releases the phosphate, so it will synthesize what? ATP. So ADP will convert into ATP. This is the thing. So from pyruvate it should form phosphoenol pyruvate. How it will form? So this pyruvate first it will convert into oxaloacetate. Remember guys, this is an substrate in what? Krebs cycle. Oxaloacetate, it's a substrate in the Krebs cycle. So again I am repeating that there is a shortcut to remove the substrates of what? Krebs cycle. That is, citrate is starting substrate for making oxaloacetate. Okay, citrate. Next is isocitrate. So starting it's citrate is Krebs starting substrate for making oxaloacetate. Citrate is isocitrate, Krebs is alpha ketoglutarate. Starting is succinyl CoA, substrate is succinate. So FAR is fumarate, making is malate, oxaloacetate is oxaloacetate. Okay, so this pyruvate will form the substrate in the Krebs cycle, that is oxaloacetate, by using an enzyme that is actually we already discussed, that pyruvate carboxylase. Whenever you see the enzyme carboxylase, remember guys, we need ABC. So it need ATP. So B, biotin prosthetic group. So next C is CO2 carboxylase. So carbon dioxide will be added to the pyruvate, so it will form oxaloacetate. Understood? ATP, it need what? ATP. The phosphate group should also added. From where it will get the phosphate group? From ATP it will convert to ADP. Understood? So CO2 will be added. Okay, finally we will get what? We will get oxaloacetate. From pyruvate to form oxaloacetate, it will utilize an enzyme that is called pyruvate carboxylase. So pyruvate carboxylase is a carboxylase enzyme. We need ABC. So we need ATP. So there is phosphate group. We will get from ATP. So ATP will be converted to ADP. So phosphate will be added to the pyruvate, form oxaloacetate. So we need biotin and also CO2. Understood? So this oxaloacetate will form phosphoenol pyruvate. Okay, like this actually pyruvate will form finally the phosphoenol pyruvate. So from this phosphoenol pyruvate, it will actually, this is only irreversible step. So phosphoenol pyruvate can form 2 phosphoglycerate by using the same enzyme. What is that? Phosphoenol pyruvate will form 2 phosphoglycerate by using the enzyme that is enolase, because enolase in the phosphoenol pyruvate, enol group is there. So from 2 phosphoglycerate it will form phosphoenol pyruvate with the help of enolase enzyme. It's an irreversible step. So 2 phosphoglycerate will form phosphoenol pyruvate by using enolase. Given from phosphoenol pyruvate, it will form 2 phosphoglycerate by using the same enzyme. Okay, so now we will talk about how the oxaloacetate will form phosphoenol pyruvate by using an enzyme, beautiful enzyme, that is phosphoenol pyruvate carboxykinase. Okay, from oxaloacetate it will form phosphoenol pyruvate by using the enzyme phosphoenol pyruvate carboxykinase. Okay, here we are using the enzyme kinase, so we need a phosphate group. From where we are getting the phosphate group? Here actually we will use GTP. Okay, this GTP will convert to GDP. From here the phosphate group will be added. From here the phosphate group will be added to the substrate to become phosphoenol pyruvate. Okay, you understood guys? So here the enzyme we are using is phosphoenol carboxykinase. So phosphate group will be added from GDP. Understood? From GTP, that GTP will be converted to GDP and the phosphate group will be added. And one more important thing is that the carbon dioxide will be released, because here in the oxaloacetate the carbon dioxide is there. Carbon dioxide will be eliminated. Okay, by this way it will form finally phosphoenol pyruvate, and from here it will enter into the reversible glycolysis pathway. Okay, reversible glycolysis pathway finally forms glucose. Okay, this is how it will form the glucose, and this is the important substrate pyruvate. Actually, how that pyruvate will form? Actually, it will come from the lactate, that we will see in the future slides. Okay, this is how glucose is synthesized from the lactate. Or we will talk about the pyruvate itself here right now. Okay, next slide. So up to here I think it is clear. Okay, when we talk about I am repeating again. So how this pyruvate will form phosphoenol pyruvate? By using two important enzymes. Okay, by using two important enzymes. So we have already talked about two enzymes. So two more important enzymes are there that will useful to reverse the three important steps or three irreversible steps in the glycolysis. Okay, one we talked about the pyruvate carboxylase enzyme, and another one we already talked one more enzyme is phosphoenol pyruvate carboxykinase. One enzyme will add CO2, one enzyme will release CO2 and add phosphate group. Understood? One enzyme will help to form the pyruvate to oxaloacetate, and another enzyme will form the oxaloacetate to phosphoenol pyruvate. Okay, this is this is how the pyruvate will form phosphoenol pyruvate. Okay, the pyruvate cannot directly form the phosphoenol pyruvate because this is a irreversible step. So, okay, to form the phosphoenol pyruvate, it take two steps. First this pyruvate will form oxaloacetate, and from this oxaloacetate it will form phosphoenol pyruvate. Okay, second step. So we have seen this next the phosphoenol pyruvate will form pyruvate, it will form 2-phosphoglycerate by using the enzyme enolase. Next, 2-phosphoglycerate will form 3-phosphoglycerate by using the enzyme phosphoglyceromutase. Okay, and this 3-phosphoglycerate will form what? 1,3-bisphosphoglycerate by using the enzyme kinase. Okay, phosphoglycerokinase. From this 1,3-bisphosphoglycerate, it will form what? Glyceraldehyde 3-phosphate by using the important enzyme that is called glyceraldehyde 3-phosphodehydrogenase. That's very important enzyme. This glyceraldehyde 3-phosphate enzyme will form dihydroxystone phosphate by using what? Which enzyme? By using isomerase enzyme, that is triose phosphotriose isomerase triose phosphoisomerase or phosphotriose isomerase. Okay, whatever the enzyme. So this two substrate, 3 carbon substrates will form directly from the 6 carbon substrate, that is fructose 1,6 phosphate, by using the enzyme aldolase. These steps all are reversible. Except phosphoenol pyruvate to pyruvate. Okay, this pyruvate cannot form back the phosphoenol pyruvate. With the help of oxaloacetate only it can form phosphoenol pyruvate. Remember guys, this is very important step. Next, the fructose 1,6 bisphosphate has to form fructose 6 phosphate. Okay, here actually another irreversible step is there. Is fructose 1,6 bisphosphate will form fructose 6 phosphate. How it forms? So this first fructose 1,6 bisphosphate should release one phosphate group. So that means it has to break phosphate bond. Okay, how it will break? So remember guys, phosphatase enzyme, fructose 1,6 bisphosphatase. Fructose 1,6 bisphosphatase is the enzyme will break what? Phosphate group. So after breaking the phosphate group in fructose 1,6 bisphosphate, one phosphate group will be released and forms fructose 6 phosphate. Understood? So finally fructose 6 phosphate form. So next step is fructose 6 phosphate will form what? Glucose 6 phosphate by using what? Isomerase enzyme. Glucose 6 phosphate, it's an isomerase, so it will form glucose 6 phosphate by using the enzyme that is isomerase. Glucose and fructose is isomerase. This is a irreversible step. And one more thing irreversible step is glucose 6 phosphate should form glucose. How? So again the same thing, there is a phosphate bond between glucose and phosphate. Phosphate. Okay, there is a phosphate bond between glucose and phosphate that should be broken by using what? Phosphatase enzyme. That is actually glucose 6 phosphatase. The 6th carbon phosphate group is attached. That phosphate bond should be broken by using phosphatase enzyme. When this happens, we will finally get glucose. Remember guys, most important thing, so we have to remember glucose 6 phosphate. This is a wonderful enzyme which is present only in where? Liver and kidney. Okay, this is only present in liver and kidney, that too especially in endoplasmic reticulum. Endoplasmic reticulum. Okay, in only endoplasmic reticulum it is present. Okay, it is not present in muscles and also adipose tissue. It is not present in muscles and adipose tissue. It is only present in the liver and kidney, especially in the endoplasmic reticulum. Understood? So there only the free glucose will be synthesized. Okay, thank you guys. And this is how actually the pyruvate will form the glucose. So actually we need to talk about what lactate forming the glucose. How the lactate forms? Actually lactate will form first what? Pyruvate. How this will happen? There is an irreversible enzyme that is called LDH, lactate dehydrogenase. This lactate dehydrogenase, it is a reversible enzyme, so it will either can form lactate to pyruvate or pyruvate to lactate. That's what we'll see in the next slide, guys. Okay. So now we'll discuss total. So what is the thing we have discussed in the previous slides? So there are 3 irreversible steps in the glycolysis pathway that should become reversible. How? By using 4 enzymes. What are those enzymes? 2 enzymes are utilized to convert the pyruvate to phosphoenol pyruvate. That is pyruvate carboxylase, and next one is phosphoenol pyruvate carbokinase. Carboxylase enzyme we need ABC. Carbokinase enzyme it will add phosphate and release CO2. Okay, so when we see the glycolysis pathway, this is the glycolysis pathway. So glucose will convert to glucose 6-phosphate. Okay, this is an irreversible step, and glucose 6-phosphate can convert it to fructose 6-phosphate. It is a reversible step. So fructose 6-phosphate to fructose 1,6-phosphate, this is irreversible step. So the enzymes will vary. Understood? Enzymes will vary in the first step and third step. Okay, and one more thing, in the last step also the enzymes will vary. Like here, this is the last step, the phosphoenol pyruvate to pyruvate. Here these steps 1, 2 and 3, these are actually irreversible step. 1, 2 and 3, these are the irreversible steps. For making these steps reversible, we need 4 enzymes. 3 irreversible steps should be made reversible by using 4 enzymes. Okay, this is the thing you have to remember. Understood? Alright, I'll go to the next slide. Okay, now we already seen there are 4 substrates in gluconeogenesis pathway. One is lactate, another one is glycerol, another one is glucogenic amino acids, and another one is fatty acid chain, that is propionyl CoA enzyme, coenzyme A. Okay, so now we'll see how the lactase is converting to glucose. Very important and very easy, guys. So we will get the questions directly. Write the glucogenic pathway from lactate to glucose. How the lactate is converting to glucose? Very, very, very easy. So we already know in the glycolysis pathway, the pyruvate have 2 fates. Pyruvate have 2 fates. One is in low glucose level and another one is high glucose level, right? We have already seen that. At the low glucose level, what will happen? This pyruvate will be converted to lactic acid by using what? It's lactate or lactic acid, both are same, दोनों एक ही है, no problem at all. So when this pyruvate is converting to lactate or lactic acid by using a beautiful enzyme that is called lactic dehydrogenase enzyme. Okay, lactic dehydrogenase enzyme, it's a reversible enzyme. It can convert the pyruvate to lactate or lactate to pyruvate, both are same. Okay, this lactic acid and lactate can be stored in liver and muscles. Okay, maximum amount it will stored in the liver and some amount it will be stored in the liver muscles. Okay, this lactic acid, okay, again can be converted in the gluconeogenesis pathway, converted into pyruvate, and this pyruvate will be converted into phosphoenol pyruvate by using what? 2 enzymes. It cannot directly form, it's a irreversible steps. First it will form the oxaloacetate and it should then form the phosphoenol pyruvate. Okay, so this is how it will form. First lactate is converted to pyruvate by using what? Lactate dehydrogenase enzymes. By using lactate dehydrogenase enzyme, I'm repeating, lactate dehydrogenase. Dehydrogenase enzyme. Whenever you see dehydrogenase enzyme, you have to in your mind something should light up. That is what? It should convert a NADP plus, NAD plus to NADH, or NADP plus to NADP to NADPH, or FAD plus to FADH2. This should happen. NAD plus to NADH, or FAD plus to FADH2, or NADP to NADP plus. This should happen. Here actually NAD plus is converting to NADH. Okay, this lactate will form pyruvate. So how this pyruvate will be converted to phosphoenol pyruvate? So this is the step. The pyruvate first should convert to oxaloacetate by using the enzyme pyruvate carboxylase. It should note ABC. ABC is nothing but ATP, biotin, CO2. Okay, then it will convert to oxaloacetate. From the oxaloacetate, phosphoenol pyruvate can be formed by using phosphoenol carboxykinase. Okay, kinase enzyme. Okay, kinase enzyme, it should add phosphate group. So finally it will form PEP, that is phosphoenol pyruvate. From here it will form 2-phosphoglycerate by using the enzyme enolase. Okay, 2-phosphoglycerate to 3-phosphoglycerate by using the enzyme mutase. Okay, 3-phosphoglycerate to 1,3-bisphosphoglycerate by using kinase, because 1 phosphate group is adding. So 1,3-bisphosphoglycerate to glycerol 3-phosphate by using the same enzyme. These are actually reversible steps. Okay, from here to here, these are the reversible step. Okay, so 1,3-bisphosphoglycerate to glycerol 3-phosphate by using glycerol 3-phosphate dehydrogenase enzyme. So dehydrogenase enzyme NAD plus to NADH. Okay, next this glycerol 3-phosphate converted to dihydroxyacetone phosphate by using the enzyme. Okay, by using the enzyme, what is it? Isomerase. Okay, these two substrates will fuse to form fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate should convert to fructose 6-phosphate. This is an irreversible step where we will use an enzyme, that is phosphatase. Okay, phosphatase will break the phosphate bond and form fructose 6-phosphate. Okay, this fructose will form glucose by using isomerase enzyme. So form this fructose 1,6-phosphate to glucose 6-phosphate, we use what? Phosphofructoisomerase as phosphohexoisomerase. Okay, I forgot to write the enzyme here. We will use isomerase enzyme here. Isomerase enzyme. Okay, we will use isomerase enzyme because fructose and glucose are both are isomerase. From this glucose 6-phosphate to glucose, here the picture lies here. Okay, this glucose 6-phosphate should form glucose by using one important enzyme, that is glucose 6-phosphatase. Phosphatase means it should break some bond, that is phosphate bond, then only it can form glucose. Important thing is that glucose 6-phosphate is the enzyme, glucose 6-phosphatase is the enzyme only present in the liver and kidney. Okay, it will present in liver and kidney, right? So now we have seen how this lactate non-carbohydrate substrate form glucose. Okay, this is how it will form, right? These steps are called glycolysis steps. These are actually reversible steps of glycolysis, and there are three irreversible step. This is irreversible step, and this is one irreversible step, and this is another irreversible step. One is how pyruvate is converting to phosphoenol pyruvate, that is irreversible. Fructose 1,6-bisphosphate to fructose 6-phosphate is also the irreversible step, and glucose 6-phosphate to glucose is also irreversible step. Okay, next we will see it's a fatty acid chain, propanyl CoA. Okay, from propanyl CoA, how glucose is synthesized? Okay, this is also a very important question. It's an you should write the glucogenic pathway of propanyl CoA. So how the this is how we have to write how the propanyl CoA will synthesize the glucose. Okay, so remember guys, this is an odd fatty acid chain. Okay, odd fatty acid chain. Remember, propane is nothing but 3 carbon, right? Propanyl coenzyme A is nothing but 3 carbon fatty acid chain here. Okay, this 3 carbon fatty acids is a odd carbon fatty acid chain. Remember, all odd carbon fatty acid chains can only form glucose. Okay, can only participate in the gluconeogenesis pathway. Even chain fatty acids cannot form glucose. Okay, even this is a starvation condition, this odd fatty acid chain only can form the glucose and participates in the gluconeogenesis pathway. Understood? How this will happen? See here. First of all, this propanyl CoA will form methyl malonyl CoA. How this will happen? By using an enzyme, propanyl CoA carboxylase enzyme. This will form what? Methyl malonyl CoA. Methyl malonyl CoA. Propanyl CoA will form methyl malonyl CoA. Right? This is a 3 carbon substrate, but here actually in methyl actually 2 more carbons are there. So in this actually this is an carboxylase reaction, so 1 more carbon will be added in the form of carbon dioxide. This is very important guys. Propanyl CoA is only 3 carbon, so here methyl group is added to the malonyl CoA, right? So 2 more carbons are added. Two more carbons are added. These carbons are added. More number of carbons are added. So how this number of carbons are added? By using CO2. Okay. So this is a carboxylase enzyme. So when there is a carboxylase enzyme, so there is a ABC. So ATP will be utilized and biotin will be there and carbon dioxide will be there. Carbon dioxide is the source for carbon here. Remember guys, this propanyl CoA will form methyl malonyl CoA. See, malanoate. So when we see the substrate in the Krebs cycle, that is malanoate. Actually, it is a derivative of the malanoate. Okay, so malanoate is a 4 carbon substrate, right? So one second. So for clarification, we can go for the Krebs cycle pathway also. So melanoid, not melanoid, it's a mallet. Mallet is a 4 carbon structure. Good, no problem. Okay. So here remember guys, the carbon number is increasing. The source is nothing but source is actually carboxylase enzyme. This carboxylase enzyme will add more number of carbons. It will form what? Methyl malonyl CoA. Methyl malonyl CoA. Okay, methyl malonyl CoA. This methyl malonyl CoA will form succinyl CoA. Right? So remember guys, this methyl malonyl CoA will form succinyl CoA. Okay, so this methyl malonyl CoA and succinyl CoA both are isomerase. Both are actually carbon translate reactions or carbon shifting reactions. Okay, this methyl malonyl CoA will form succinyl CoA by using an enzyme that is called mutase. Here actually methyl malonyl coenzyme A will be used as an enzyme. Okay, important thing is that propanyl CoA will form methyl malonyl CoA by using an enzyme, carboxylase enzyme. Here this methyl malonyl CoA will form succinyl CoA by using an enzyme mutase. So what is that mutase enzyme? That is methyl malonyl CoA mutase enzyme. Okay, this enzyme requires B12 B12 as a prosthetic group. Okay, then only we will form what? Succinyl CoA. This succinyl CoA is then substrate in the TCA cycle. So remember guys, this succinyl CoA will form what? succinate, and from succinate it will form fumarate, and from fumarate it will form malate, then it will form oxaloacetate, right? From oxaloacetate it will form finally phosphoenol pyruvate, then it will enter into the glycolysis pathway, and finally it will synthesize glucose. Okay, this is very important pathway, guys. So remember, we need to know two important steps here. Okay, first is propanol CoA will form methyl malonyl CoA by using the enzyme carboxylase, that is propanol coenzyme carboxylase. This methyl malonyl CoA will form succinyl CoA by using enzyme called mutase. Okay, this is a mutation reaction because carbon shuffling reaction is taking place, so it is called actually what? Mutase. So one carbon to another carbon, the groups will be shifted. Okay, from succinyl CoA it will enter into the TCA cycle and form a substrate called oxaloacetate. So in the last pathway you have seen this oxaloacetate will form phosphoenol pyruvate by using excuse me, by using the enzyme that is phosphoenol pyruvate carboxykinase. Okay, then this phosphoenol pyruvate will form all substrates in the glycolysis pathway, finally it will form glucose. Okay, this is very important pathway, guys. Remember how to synthesize glucose from propanol CoA. It's or they can also ask how glucose is synthesized from odd fatty acid chains, or write about the pathway of gluconeogenesis by using odd fatty acid chain. So this is also most important question that usually asked in the board exams. Okay, next thing is that how glycerol will be utilized to form glucose. Glycerol is also a non-carbohydrate which is used to synthesize what? Glucose. Okay, in the body. Actually, how it will form? Glycerol first will form glycerol or you see here the glycerol can enter, it's a 3 carbon, it can form glyceraldehyde 3 phosphate. How this glycerol will form glyceraldehyde 3 phosphate? First of all, it will form glycerol 3 phosphate. How? Glycerol kinase, because this glycerol phosphate group is added, so by using ATP, right, it will form glycerol 3 phosphate. This glycerol 3 phosphate by using dehydrogenase enzyme, that is glycerol 3 phosphate dehydrogenase enzyme, it form dihydroxyacetone phosphate. Okay, this dihydroxyacetone phosphate will be converted to glyceraldehyde 3 phosphate by using the enzyme isomerase or triose phosphoisomerase or triose isomerase enzyme. Okay, this by using aldolase enzyme will be fused to form fructose 1,6 phosphate. Okay, same thing, so we'll be repeating again. Okay, so remember, guys, first of all, with glycerol will convert to glycerol 3 phosphate, so one phosphate group is adding here, so we'll use the enzyme that is called kinase. What kinase? This is actually to the glycerol it is adding phosphate group, so it is called glycerol kinase. From where we are getting that phosphate group? From ATP. ATP is converting to ADP. Okay, next this glycerol 3 phosphate is converting to dihydroxy, that means hydrogen is adding, dihydroxyacetone phosphate, hydrogen is adding. So what is the enzyme we are using here? Glycerol 3 phosphate dehydrogenase. Okay, so from where it is getting the removing the hydrogen, so it is removing from the glyceraldehyde 3 phosphate and adding to the NADH. Okay, so it is getting reduced and finally forms what? Dihydroxyacetone phosphate. This dihydroxyacetone phosphate by isomerase it forms a glyceraldehyde 3 phosphate. Finally it will enter into the glycolysis pathway, forms fructose 1,6 phosphate. So this remaining thing is same as it is. Okay. Next important thing is that glucogenic amino acids. How these glucogenic amino acids form glucose? Remember, guys, there are many amino acids which can form substrates in the Krebs cycle. When it enters into the Krebs cycle, automatically it can enter into the glycolysis. The cycle will be reversed and it will enter into the glycolysis pathway and it can form what? Glucose. Okay, remember, guys, I already discussed there are many amino acids all amino acids except leucine. It's an leucine actually exclusively ketogenic amino acids. It will form ketone bodies. Other than that, all amino acids are glucogenic amino acids. Most importantly, alanine most commonly will undergo gluconeogenesis. Okay, if you talk about glucogenic amino acids, especially we will talk about the alanine. It is frequently will undergo gluconeogenesis and forms glucose. Understood? So most frequently undergoing gluconeogenic pathway undergoing amino acid is alanine. It's a very important thing for the GPAT and many competitive exams. Alanine is an important glucogenic amino acids. Frequently it undergo gluconeogenesis and will form what? Glucose. Right? How? We'll see here. See, so there are, remember guys, this is a very important thing. This amino acids will form a intermediate in the TCA cycle. Okay, this substance in the TCA cycle will enter into the glycolysis pathway and forms amino acids will form intermediates in the TCA cycle and Krebs cycle and enters into the glycolysis pathway and forms glucose. So which amino acids will form intermediates, that we have to remember. Okay. Like if you talk about alanine, serine, glycine and cysteine, this will form pyruvate. Guys, you have to remember the amino acids. Okay, and the substrate it forms in the TCA cycle, that is Krebs cycle. Okay, alanine, serine and glycine and cysteine, this will form pyruvate. If you could talk about glutamate, glutamine and proline, histidine, it will form alpha ketoglutarate. Okay, valine, methionine, isoleucine and threonine will form succinyl CoA, and tyrosine and phenylalanine will form fumarate. Aspartate and asparagine will form oxaloacetate. Okay, for example, if you take your easy understanding, if you take aspartate and asparagine, this will form oxaloacetate. So oxaloacetate. So when we see the Krebs cycle, so where is the oxaloacetate? Here guys. This is actually oxalated. Sorry guys. Okay, sorry. This is what? Oxaloacetate. So what it will do? This oxaloacetate will form pyruvate by using what? By using an enzyme that is called pyruvate dehydrogenase. Okay, by using the enzyme that is pyruvate dehydrogenase and forms pyruvate. This pyruvate will form what? Phosphoenol pyruvate. This phosphoenol how this pyruvate will form phosphoenol pyruvate? With the help of oxaloacetate, right? Or this oxaloacetate will directly form what? Phosphoenol pyruvate, right? This phosphoenol pyruvate, I'm sorry, I discussed wrong. So this whatever the amino acids we have seen in the previous slides, for example, if you talk about asparagine and aspartate, these amino acids will form oxaloacetate, right? Okay, if you talk about oxaloacetate, this oxaloacetate will do? This oxaloacetate will form phosphoenol pyruvate. This phosphoenol pyruvate will form 2-phosphoglycerate. Okay, this 2-phosphoglycerate will form 3-phosphoglycerate. Okay, like that it will finally form what? It will finally form glucose. Okay, so this is how this gluconeogenic or glucogenic amino acids will form glucose. Okay, whatever the substrates it is forming, whatever the amino acid is forming the substrates in the Krebs cycle, it will enter into the glycolysis pathway. Okay, by oxaloacetate. For example, if some substrate is forming succinyl CoA, this succinyl CoA will form succinate, then this succinate will form fumarate finally will form oxaloacetate, and this oxaloacetate will form phosphoenol pyruvate. Okay, this phosphoenol pyruvate will enter into the glycolysis pathway, right? Some substrates are forming alpha ketoglutarate. Okay, sorry, some substrates are forming alpha ketoglutarate. Okay, some substrates are forming alpha ketoglutarate. This alpha ketoglutarate will form succinyl CoA, then succinate, then fumarate, then malate, then oxaloacetate, then finally it will enter into glycolysis with the help of phosphoenol pyruvate. This is how this sorry guys, this is how this glucogenic amino acids will form glucose. Okay, or glucogenic amino acids will enter into the Krebs cycle and form glucose. Okay, this is how this non-carbohydrates this is how this non-carbohydrate sources will also utilize to form glucose, and this will act as an important source for glucose during starvation condition. Thank you guys, have a nice day.